It's nice to be with you again. Welcome back to your class microbiology, and in this meeting we are going to discuss bacterial metabolism. Under bacterial metabolism, we're going to have a review of your enzymes and energy in metabolism, anaerobic and aerobic respiration, glycolysis, Embden-Meyerhof pathway, and fermentation. I'm sure you are not new to these topics. If you have finished your biochem, you have discussed enzymes and also likewise glycolysis and respiration, and also with your citric acid cycle or the Embden-Meyerhof pathway. So all of these are needed background of information in order to understand fermentation. So what is metabolism? Metabolism is the sum total of the chemical reactions in an organism, and there are two types. You have the catabolism and the anabolism. For catabolism, uh, it provides energy. It is the mechanism that builds blocks for anabolism. So in terms of anabolism, the keyword here as contrast to catabolism is in anabolism, uh, it is a metabolism that uses energy and building blocks to build large molecules. So in the case of catabolism, the keyword here as contrast to anabolism is it provides energy. Whereas for the case of anabolism, it is the uses energy as the keyword as contrast to the other type of metabolism. And then let us start discuss or have a review on enzyme. Enzymes are biological catalysts. Enzymes either lower the activation of energy required for biological reaction to occur, and also likewise enzymes could be categorized as protein. They have their own specifics for the reaction they catalyze. They are not used up in the reaction to remain as enzyme. They speed up reaction in living system, and categorically they all end with the suffix "ase". Now, why do we call enzymes as protein? Apoenzymes are protein part. Enzymes are cofactor, and as a cofactor, they are non-protein part, but rather cofactors are often metal ions. Coenzymes are organic cofactor. Another type of enzyme, holoenzyme, is formed together by apoenzyme and coenzyme or a cofactor. Now, what are the energy requirements? As we are trying to discuss these things, I would like you to have a mindset on our discussion about the nutritional requirement of microorganisms for growth and development. I would like you I would like to require you to have uh, a recall on our discussion on bacterial nutrition. So in terms of reactants without enzyme follows this dark green line. Whereas for B reactant, reaction with enzyme follow this line. So in terms of energy requirement, A reaction with enzyme, the activation energy with enzyme could only be as big as this. But activation energy without enzyme follows this line. So in terms of a final requirement, enzymes are more self-sufficient rather than energy production without enzyme. And then haloenzyme. In our discussion in biochemistry, we have apoenzyme as defined as the protein portion, and we have the cofactor and being identified as the non-protein portion. So as a protein portion, the apoenzyme is inactive, whereas for the case of a coenzyme, they are the cofactor and they serve as activator. So the coenzyme helps the apoenzyme in order to produce a substrate and the enzyme reaction. So if this portion is the apoenzyme, with the help of a coenzyme, it will be able to read this portion of the substrate. And imagine that you do not have the coenzyme and the substrates would like to work with the holoenzyme here. For sure, the, the ending here does not suit the shape or the characteristics embedded in this portion of the holoenzyme. Now, what are coenzymes? Coenzymes are organic molecules. They are derived from vitamins. And some coenzymes act as electron carriers in metabolic activities. In biology, we have learned that there are two important coenzymes in metabolism. They are the NAD or the nicotinamide adenine dinucleotide and the FAB, which is the flavin adenine dinucleotide. Okay, in a macro level, you have here more illustrations as to how the enzyme substrate work. So here is the substrate. This is the substrate. And then we need this is the enzyme. And then the substrate is the pink one. So this is the enzyme. And you have the substrate here. If the substrate fixed with the shape or character or nature of the enzyme, then it could further undergo reaction, thus forming this product. And if the product again is in conformity with the nature of the enzyme, then the reproduction or the growth or the reaction will push through. So in this illustration, you have there the substrate working with the enzyme. So since it fits very well, then it could produce another substrate that will work with another enzyme. Okay, these are the factors that influence enzymatic activity, as if they are similar with that of bacterial growth and nutrition. So there is an optimum temperature. So as the energy or temperature increases, it could reach a certain peak as the optimum level or the log phase, and then eventually the growth will go down as the temperature increases. And then what is the effect to enzyme and the pH value? There are some enzymes that could reach at a high level. However, there is always what we call the optimum level or the lag phase, and then followed by the decreasing phase or even death phase. This is also true with the substrate. There's always a peak that influence the activity of the enzyme. So you have temperature, the pH, the substrate concentration, and of course we have the inhibition. Now, how does inhibition works? You have the competitive inhibitor, or we have competitive inhibition, whereby the inhibitor looks like the natural substrate and fills the active site, preventing the enzymatic reaction. And we have also the non-competitive inhibition. It follows and locates for the allosteric sites which inhibit the binding to another site of or lead to another site on the enzyme. So this is the normal binding substrate, the one that we were discussing. And we have also inhibitor, the presence of an inhibitor. So in order for this to go here, what will happen is to remove the inhibitor and find another place of the substrate or the enzyme rather, in order for the substrate to fix itself in the active site. So that is how the inhibitors also works. Okay, all of these are responsible in the formation of energy or adenosine triphosphate. So how could that happen? Again, going back to your biochem, adenosine diphosphate plus another molecule of phosphate with the presence of energy will now form the diphosphate into triphosphate. So first, you have to produce the ADP, and then next you provide the phosphate, and then add energy, and finally adenosine triphosphate is produced. Now, where is phosphorylation or where is energy? Through the process of phosphorylation, that is adding phosphate to ADP to make ATP, energy is produced. Cells can do this in two ways. So as with microorganism as a unicellular organism. First is the substrate phosphorylation. The phosphate is transferred directly to the ADP to form ATP. Another is oxidative phosphorylation. So electron carriers such as NAD, nicotinamide adenine dinucleotide, plus transfer electrons to a series of carriers in the electron transport chain where energy is released and transferred to ADP to make ATP. So it's a longer process as compared with the substrate level phosphorylation. So if we're going to look at your metabolism, anabolism plus catabolism, as shown in photosynthesis, it requires respiration. As shown also in respiration, it requires photosynthesis. Energy production is equal to energy consumption. This is a thing that you have learned in the conservation of mass and energy. So if we're going to look at this illustration, we have big molecules like protein, starch. The protein after catabolism will produce smaller molecules. In the case of protein, as large molecules, catabolism happened and then amino acids will be produced. So the presence of the amino acids now will provide the presence of energy. However, it could follow and reverse the reaction, and that is anabolism. From the smaller amino acids, they are brought together to form larger molecule, and that is protein. And then this is your illustration of catabolism using your energy derived from the three big molecules, protein, carbohydrates, and lipids. So for the case of protein, there should be a breakdown of amino acids, but for the case of lipids, there should be a breakdown in order to form fatty acid. For the case of carbohydrates, in the form of glucose. So amino acids plus sugar plus fatty acids with the presence of glyceraldehyde 3 phosphate, pyruvic acid undergoing glycolysis will produce acetyl-CoA. And this acetyl-CoA is one of the initial things needed in Krebs cycle. So next, this is a micro illustration as to how nutrient biosynthesis is happening within the cell. So the nutrient for biosynthesis via anabolism enters the cell in order to provide raw material for the utilization of energy through the process of catabolism like chemical light energy. So this one now, catabolism will be the one directly can feed the patients. So that is what you call catabolism. And the cellular respiration is just the reverse of uh, cellular metabolism. Again, you have seen this illustration in your biochem. So going back, here is the difference between aerobic and anaerobic metabolism. So to illustrate the aerobic mechanism, you have here the glycolysis. So the glucose will be converted into NADH to FADH. In this portion, the release of carbon dioxide is shown in this manner, and at the same time the formation of adenosine triphosphate. So there are four subdivision of cellular respiration, and all of these are undertaken by your microorganisms. So you have the glycolysis, and then Krebs cycle, the electron transport system, and of course the chemiosmosis. Now let us start to look into glycolysis. Glycolysis is simply defined in your younger years as glucose burning. So glycolysis from your carbohydrates occurs either in the presence or absence of oxygen and yields relatively little adenosine triphosphate. Most living cells uses this process, including your microorganism. So here are your different stages of glycolysis. Of course, you have here the molecular carbohydrates, and then followed by the conversion of ATP to ADP. Then glucose 6-phosphate is produced, and then the ATP will again broken down into ADP. Then you have the fructose 6-phosphate to fructose 1 diphosphate reaction or conversion. And then finally, it will produce your um, dihydroxyacetone or DHAP and your glyceraldehyde phosphate. And then we have also the after the preparatory stage, then energy conserving stage will follow. For a series of ADP to ATPs release. And then fermentation occurs in the absence of oxygen. It produces a small amount of ATP and the organic molecule, but not the oxygen, is the final electron acceptor. End product are alcohol and in some cases, if it's not successful, acids are produced. So you could just simply try to imagine again the formation of our basi wine and basi vinegar. So this is a schematic diagram for your fermentation. This is just a review. I am sure you can still recall this illustration as presented in your biochem. So we have the citric acid cycle or the Krebs cycle. You have aerobic process. Krebs cycle has the potential to generate most energy for cells. And the product for this cycle are uh, assumed. From the three molecules of NADH to one molecule of FADH and one molecule of ATP. Why times 2? Because glycolysis generates two molecules of pyruvic acid at the end of the cycle. So going back, so you have here the different substrates that are used to reproduce and the different enzymes responsible. So here are your end products of pyruvic acid vis-a-vis what kind of organism. So in the case of Streptococcus lactobacillus and Bacillus per se, the product of fermentation is lactic acid. And then for Saccharomyces, ethanol or carbon dioxide. That is why yeast is needed in the process of baking. In the case of Streptococcus and other bacillus, they are needed in the fermentation of lactic acid, especially in the formation of cheese and other dairy products. And then you have Propionibacterium. They are responsible in the production of propionic acid needed in the formation of tasty or sour vinegar. And then you have also Clostridium producing some solvents like butanol, acetone, isopropyl alcohol. And then for the case of Escherichia coli is found in as enteric organism, and these are the possible byproducts. But these are not, of course, commercially used, but rather the presence of this implies the presence of GIT infection. And then the same true with the case of Enterobacter, other Enterobacter. So here is a simple procedure in making wine. You need any source of sugar or carbohydrates, yeast as the source of your microorganism that will ferment the carbohydrates. So it does not require energy or does not require oxygen rather. So what is happening in the bottle? It is fermentation. So this is all for this meeting. Then we will be discussing the other part of metabolism.